So welcome everyone to the 2022 Bell Lecture. The aim of the Bell Lectures is to bring world-class physicists to McGill to tell us about exciting science and inspire the next generation. This lecture series is made possible partially by an endowment from the Bell family in memory of Robert Edward Bell, Bob Bell to his friends and colleagues, who was a distinguished member of the department. Bob Bell passed away exactly 30 years ago today, on April 1st, 1992, at the age of 72. Bob Bell's illustrious career spanned excellence in science as well as academic administration, thus epitomizing the McGill's idea of excellence in scholarship and services. Bob Bell was principal and vice chancellor of McGill from 1970 to 1979, which was a tumultuous period of time. During his 9-year tenure in the post, he restored harmony, trust, and financial solvency to the campus and was much respected by everyone. Bob Bell was a world-renowned nuclear experimentalist, famous for his measurement of deuteron binding energy and the discovery of a new form of radioactivity called the delayed proton radioactivity. This was the third such discoveries at McGill, the other two being alpha and beta rays by Rutherford. Bob Bell's scientific contribution was recognized by many honors he received, including the Fellow of the Royal Society of Canada, Rutherford Chair, and the Companion of the Order of Canada, and many more. I believe it is fitting tribute to Bob Bell's legacy that we have Professor Taekjip from Johns Hopkins University today, who will deliver the 2022 Bell Lecture. I will now turn the floor to Paul Wiseman, who will introduce our distinguished speaker. Paul.
Thanks, Sang-Yong, and welcome everybody to the 2022 Bell Lecture in Physics. It gives me immense pleasure to introduce Taekjip Professor Ha, joining us as Thomas Jenkins, Department of Biophysics, Johns Hopkins University. I'm very excited that TJ could join us at McGill to give the Bell Lecture because Professor Ha is one of the preeminent single molecule experimental biophysicists, and I'm very much looking forward to hearing his talk today. Briefly, I'd like to give you a little bit in terms of his academic pedigree and background. Professor Ha did his Bachelor of Science in Physics at Seoul National University, then went on to work on his PhD in Physics at UC Berkeley, where he worked with Shimon Weiss and Daniel Chemla. He moved on to do 1 year of postdoctoral fellowship with Shimon Weiss and Daniel Chemla at Lawrence Berkeley National Lab, and then went to continue in terms as a postdoctoral fellow with Nobel laureate Stephen Chu at Stanford University. In 2004, TJ was appointed assistant professor of physics and biophysics at University of Illinois Urbana-Champaign, and he quickly moved up the ranks, soon becoming associate and then distinguished full professor at University of Illinois, where he was one of the leading figures in terms of establishing Urbana-Champaign as one of the key centers of biophysics in the United States and certainly the world. In 2015, TJ moved to Johns Hopkins University, where he became the Bloomberg Distinguished Professor of Biophysics and Biophysical Chemistry and Biomedical Engineering at Johns Hopkins University, where he remains today. I would just like to note some of his many awards, too many to list, but I want to point out early on, Fluorescence Young Investigator Award from Biophysical Society, Alfred P. Sloan Fellow. He was elected Fellow of the American Physical Society in 2005. 2015, elected member of the National Academy of Sciences. 2018, the Kinoshita Award in Single Molecule Biophysics, Biophysical Society. This is a particularly important award in terms of single molecule biophysics in the memory of Professor Kinoshita. and in 2021 elected member of the National Academy of Medicine in the United States. This is just a sampling of this. I would also point out that he was elected president of the Biophysical Society in 2021. It's on various reviewing boards and editorial boards, but I will just say, I think we'll turn it over to TJ to hear about the exciting science that he's going to present us in the Bell Lecture today. Thank you, TJ, for agreeing to give this. Very much looking forward to it. I should point out that I will be monitoring the chat for questions. I think TJ has a pause point partway through his lecture, so we can pause to take some questions. I will ask people to unmute and turn on the camera if they feel comfortable in asking a question. Otherwise, I can read it from the chat and we'll just work it out as the case may be. And of course, we'll finish with questions at the end. So without further ado, turn it over to Professor Ha. Very much welcome you for the Bell Lecture 2022.
Thank you very much, Paul, for the kind introduction and also invitation, and also Professor John for telling us all about Dr. Robert Bell, that after whom the lecture is named. So it is my great honor to speak in the colloquium, especially as one of the Bell lecturers in the series. After I moved to Johns Hopkins now 7 years ago, I learned that the campus mascot is a blue jay. So in my talk here, I'm going to highlight a word that I want to highlight the most in each slide's title using the blue jay color. So if you get lost, if you get bored, you can just play a game asking yourself, okay, why did the speaker pick that particular word and highlight in this slide, perhaps. So here I'm highlighting a double here. So I think it's this first year of Bush administration, the second Bush administration in 2001. He appointed Elias Zerhouni as a director of the U.S. National Institute of Health. And one quote that I remember from reading the announcement is that he said that understanding life is the biggest challenge ever faced by mankind. It'll define this current century. So my thought back then, maybe still is that this means that understanding life is too important to be left to biologists alone. And that's why I think people who are trained outside biology should also jump in to help out. So DNA is an iconic molecule. Top left is plate structure found in the Hill of Berkeley, Lawrence Hall of Science. And some people like DNA enough to make a tattoo. I guess I don't. And you can also wonder what happens if you climb the DNA stairways. And this is a photo of Watson and Crick posing in front of a camera, trying to reenact the discussion about their proposed structure of the double helical structure of the DNA. So this is a double helical structure of the DNA drawn by Crick's wife on their kitchen table for their Nature paper in 1953. Two strands winding around each other to form the double helical structure. And that paper ended with this with a great quote. It has not escaped our notice that the specific pairing suggests a possible copying mechanism for the genetic material. And such a modest statement that some people call it the understatement of the century, the previous century. And in fact, they got worried that someone else may propose this in a more explicit manner and take the credit and the big prize. So months later, they published another paper proposing this base pairing between bases A and T and C and G as the explicit mechanism, and the rest became history, along with the big prize. So in terms of physical dimensions, there are some useful numbers here. So diameter of the double helix about 20 angstroms at 2 nanometers. And each base pair shown by the horizontal line is separated from the next one by 3.4 angstroms. So basically one base pair gives you about 3 angstroms of distance. And you get one full turn after about 10 base pairs. So that is the kind of dimension you can think about. We also know that on the backbone of the DNA, you have a negatively charged phosphate groups. So and they are really just a few angstroms of each other. So it's highly charged negatively, and you can call it an electrostatic monster. So you need to have some ions to neutralize the repulsion between the charges to allow DNA to form a double helical structure. DNA is very flexible. So here's a figure from an old paper by the Stephen Chu's lab about, I would say about 30 years ago, not quite 30 years ago. So they had long DNA, fully stretched about 30 micrometers long, and then they can stain the DNA with the fluorescent dye. You can actually visualize the DNA fully stretched. And they attach one bead to the end of the DNA, and then you can trap it using optical traps. And then when you apply flow, then you can stretch the DNA by a hydrodynamic drag. And then at t equals 0, you turn up the flow, and DNA will relax to form this more collapsed structure. And you can measure how quickly the relaxation occurs, and so on. So that is about using DNA as a single polymer to study polymer physics. So DNA is quite flexible at that length scale. You can actually measure that flexibility in a more rigorous way by applying force to the end of the DNA as a polymer, and then measure the extension. The squares are symbols denoting the experimental data points. As you increase the force over more than 2 orders of magnitude, you can see extension increases. And remarkably, you can use a very simple model called the worm-like chain model to fit the data quite nicely. This is a solid line that is a fit. And so that was remarkable because the model, as I explain next, does not take into account DNA sequence or any of its chemical structures. But nevertheless, it works really well over several decades of the force range. So what is the worm-like chain model? Mathematically, if you imagine a flexible polymer and have a coordinate running along the DNA polymer as following the number S here, that if you have one point S equals 0 and then another point S, you define tangential vectors, unit vectors, and then take the inner product. And then if you take the average of all possible polymer configurations, then that should decay exponentially over distance S along the polymer. And the decay length scale is called the persistence length, and is for double-stranded DNA about 50 nanometers, so 500 angstroms. And because one base pair is about 3.4 angstroms, so that corresponds to about 150 base pairs. So what it means is that DNA is pretty stiff on a shorter length scale, but once you go beyond 150 base pairs of 50 nanometers along the DNA, then it forgets this original orientation. So it becomes highly disordered by beyond that length scale. Another quantity that we can define to quantify the DNA flexibility is called the J factor, and it's an effective concentration of one end of the polymer around the other end of the polymer. So for example, if you imagine a worm here and then you use a knife to cut the worm into a very small piece, then once you make the piece short enough, then the one end will actually never see the other end because it is actually quite rigid at that length scale. So this J factor will become vanishingly small as you make the DNA shorter and shorter. So you expect to see a major decrease occurring around 150 base pairs, and that's exactly what this mathematical model calculation shows. And the oscillation here is due to the 10 base pair periodicity of the double helical structure. So that's what you expect from a worm-like chain model. But if you look at DNA in nature, you can see very frequently DNA that is tightly bent. In a virus capsid, you can actually package a DNA very densely into a small volume of only about 30 nanometers across. That's even shorter than the persistence length of 50 nanometers. But DNA can be sharply bent in this small space. And there are many DNA structures bound to various proteins where DNA is sharply bent. So you have here's a 10 nanometer diameter circle. And here's another structure that I'll mention later, where DNA is wrapped around a protein core a couple of times. And then again, the diameter of 10 nanometers. And that corresponds to about 100 base pairs of DNA forming a circle of that diameter. So if you actually try to calculate the energetic cost to make DNA circle of that diameter, and that's the formula. And in KT, thermal energy relative to if you calculate how many base pairs of DNA to form a circle, so if you put 100 base pairs here, then you get about 30 KT. So 30 KT is a really large number, and if you're trying to wait for spontaneous fluctuations to give you that much fluctuation in thermal energy, you'll have to wait for a long time. So that is interesting. DNA is very rigid at that length scale according to the worm-like chain model, but nevertheless, proteins are able to bend the DNA very sharply. So that is a kind of a puzzle. So my former student Reza, shown here, when he was a grad student, he developed a protein-free single molecule method to measure DNA flexibility at a very short length scale, and they found that DNA is actually quite flexible, extremely flexible at that length scale compared to what you expect. So let me explain what that method is. So we are using a method called FRET, it's fluorescence resonance energy transfer. So you have a fluorescent molecule in green. We call that donor. And then if you excite the molecule using a laser, let's say blue color, and you get a photon coming out at a lower energy, so green photon, for example. But if you have a second molecule nearby within just a few nanometers of each other, then excitation can be directly transferred to the acceptor without involving an actual photon emission. And then you can excite the acceptor, a red molecule, which then emits a red photon. So you excite the green molecule, but you get signal from the nearby red molecule. So you can ask, okay, how many red photons would you get if I excite the green molecule 100 times? And that's the definition of energy transfer efficiency, or FRET efficiency, is given by this expression where R is a distance between the two molecules. And then R0 is the relevant length scale, typically around 3 to 7 nanometers, depending on a particular pair of the dyes that we use. And this 6 power here comes from dipole-dipole interactions between the two molecules in Fermi's golden rule. And so in terms of actual signal that we get from a molecule, if the molecules are very close to each other, you get mainly the red signal coming from the pair. When they're far away from each other, mainly green signal. And at the exact distance about 0, you get an equal signal from both molecules, half and half. So if you actually measure the intensities of a green and red signals, you can actually deduce the distance between the two at the particular point of measurement. So here's an illustration for those who like watching tennis players. So here, imagine having two dimolecules at the two hands of this tennis player, and the backswing, two are far from each other, so you get mainly green signal that is directly excited by the laser. But after follows through, then you get a small separation, and there's a strong energy transfer. You get many signal from the red molecule. So Fred is a really nice single molecule spectroscopy ruler. So how does a single molecule assay work? So here we have a DNA molecule that is tethered to a surface using a well-defined molecular glue, and then we attach the green and red molecules to the overhangs of the DNA that are single-stranded, and they are complementary to each other in terms of sequence, so that if you go from low salt conditions to high salt conditions, you add some ions to allow the two ends to find each other and then base pair with each other. Then you form a circle, and that circle formation will result in decrease in distance between the two molecules and increase in Fred. So you can see that green signal will suddenly go down when circle forms, and red signal will go up at the same time in an anti-correlate manner. And you can also calculate the Fred efficiency in blue, and then you can see the beautiful signal when that circle formation occurs. So my student Raja decided to perform this measurement now using many different lengths of the DNA shown here. And then so measuring the kinetics of circle formation, looped fractional loop molecules as a function of time. And for one particular length, he saw that it takes about 15 minutes to form a circle on average. And you can actually plot the looping time as a function of DNA length, and then see that there is some variation, maybe it's even some oscillation that can be seen. But as you go from 200 to 100 to 70 base pair long DNA, we see only a modest increase in the looping time. So it slows down, but not all that much. Whereas according to the one-leg chain model, the J factor, for example, should drop tremendously over this length scale. So that was surprising, and we have done some testing to rule out the possibility that some other things that we do, such as surface tethering and modification, is affecting the result, and we actually get the same exact result when we bypass the surface tethering. So we can actually try to convert our data into the J factor and compare to the Ohm-like chain model, and we see a really large deviation even when you allow very generous boundary conditions. Even in the worst case, this or best case for the model, that's the dotted line, our data actually are still off by 100 to 1,000-fold from the prediction of the Ohm-like chain model. So whereas if you go to it's just all right. So when we go to the 200 base pair long DNA, then there's a pretty good agreement. So when you go to a very short DNA, then DNA appears to be much more flexible than what we expected. So in the end, maybe it's not so surprising that proteins can bend the DNA so sharply, because DNA itself at that length scale can bend spontaneously very well. So what about its implications in biology? So in our cell and human cells, DNA is very long. Eventually gets compacted into chromosomes shown here during the cell division, but it occurs through multiple layers of compaction. And the initial layer of compaction is formation into a structure called the nucleosome. In the nucleosome, you have 147 bases of DNA wrapped around the protein core, shown in gray, about 1.7 turns. So many years ago, we decided to combine single molecule FRET measurements that we do with optical tweezers to measure conformational changes of a single molecule using FRET, but as a function of force that you apply using optical tweezers. And wanted to study using this the mechanical deformations of the nucleosome. In a previous study published here by another lab, they use optical tweezers to apply force or tension to the end of the DNA wrapped around the protein core in a nucleosome, and then measured the DNA unwrapping and showed that it occurs in multiple stages. So at low forces, you unwrapped the outer turn of the DNA, and as you increase the force, you unwrap the inner turn, and at even higher forces, protein core can be ejected and becomes irreversible. And that actually measurement made an assumption that unwrapping occurs in a symmetric manner from both sides together. So we wanted to test that idea by putting the dye on the DNA, green dye in the middle of the DNA, and then red one at the entry point, so that you can see a major increase in distance, a major decrease in energy transfer efficiency upon outer turn unwrapping. And you may get additional distance increase if you unwrap the inner turn. And here we can try to measure local changes explicitly from one side of the DNA. So we have a optically trapped bead, and then the long DNA linker linked to our nucleosome with fluorescent labels, and then tether it to a surface, and then you can detect fluorescence using focused laser beam in a confocal fluorescence microscope. So here's the data. So if you use the standard probe set that we have mentioned to you, we call outer turn probe by putting the dyes so that they are close to each other in an intact structure, and then you should see a large decrease in FRET as you unwrap the outer turn. So in fact, if you plot this energy transfer efficiency or proximity factor between the two dyes as a function of force, as you increase the force, you see a major reduction at around 2 to 3 piconewtons of force. So that's the low force, and then you can unwrap the outer turn. But if you move the probes to the bottom of the structure, so that you need to unwrap at least 20 to 30 base pairs of DNA before you see decrease in energy transfer, increase in distance, you have to go to much higher forces to see DNA unwrap. And this is matching the previous model that at low forces you see outer turn unwrap and higher forces inner turn unwrap. We made a mistake of doing actually one more experiment, so it became a much longer project. So we said, well, if that's the correct interpretation, then we can move the probes to the other entry point of the structure, then you should also see thread go down at a low force. But instead, we found that this unwrapping signal actually occurs at a higher force. If any, it's even higher than the inner turn probe configuration. So assuming this is correct, it suggests that DNA unwrapping under tension is asymmetric, and it may even be directional. You can start from one end and propagate around the structure until you reach the other end. And so instead of inner turn followed by outer turn, I say outer turn followed by inner turn, maybe we unwrap it in a directional manner. So there are many things that we had to check, but in the end, what really convinced us and clinched the story was when my student Tui, shown here, performed additional measurements on additional positions of the dye pair. So she went around the nucleosome, performed 9 different sets of experiment. When she found that on one side of the nucleosome, unwrapping occurs at low forces, but at the other side, unwrapping occurs at high forces, suggesting that indeed there is a weak side, a weak half, and then strong half in the same nucleosome. So what is causing that asymmetry? We think that this is due to asymmetry due to all of the local DNA flexibility. So here's the idea. So if you divide the DNA that we use into a strong side and the weak side, and our idea is that strong is strong because the DNA there is more flexible and vice versa. That's because in a nucleosome structure, DNA is sharply bent. So, so if the DNA sequence make DNA more bendable or more flexible, then that sharply bent structure can be sustained even under tension that is trying to unwrap the structure. So, so that is the idea. Strong half is intrinsically more bendable or flexible. But to test this idea, you have to be able to measure flexibility of half the length of the DNA here, about 75 base pairs of DNA. And luckily, we had an essay that Reza had developed in the labs a few years earlier. Actually, in this photo from his faculty website, Northwestern, he now looks much older and looks more serious. So basically, we were able to perform the same experiment on the weak and strong side of the nucleosomal DNA. And indeed, we were able to show that strong half of the DNA is much more bendable. It forms a circle 6 times faster than the weak half. And then we have also performed additional experiments, swapping different segments and changing other factors. And that relation between DNA bendability, flexibility, and nucleosome stability was very robust. So the idea here is that in the cell, if one end of the DNA in a nucleosome structure is more flexible, more bendable, then that is held tight into the protein core, so that proteins that want to bind the DNA sequence there would not be able to bind very well. Protein will get rejected. But on the other side, if the binding sequence is on the less flexible side, then that side unwraps more readily, and protein can actually recognize the sequence more readily. So we think this is one way of actually using the DNA physics, sequence-dependent DNA physics, to control biological functions. So the next questions are, what sequence features determine DNA flexibility? And is the evolutionary signature in the genome? If this is important for function, we should be able to see that signature in the present day genome. So for that, my former postdoc Akash has developed a high throughput method that we call loop-seq. If you want DNA physics meets next-gen sequencing to measure DNA bendability, flexibility from thousands of sequences in parallel. The concept is shown here. I'm not going to show you any data today, but imagine having 4 different sequences in your library or sample, each having 10 copies. And then you give them a minute to form a loop, and by adding high sort. And DNA 1 that is most flexible will form a circle very quickly. So 9 of them will form a circle, and then 1 will not form a circle. And DNA 4, the least flexible, most rigid one, will remain mainly linear. And then we can use enzymes that can digest only the linear DNA molecules, and then keep only the circular one. In this case, the most flexible sequence will survive the process better. And then we can use next generation sequencing methods to measure the survival probability. And then we simply take the natural log of that as the flexibility of each sequence. So Akash was able to do this from more than nowadays more than 1 million different sequences. And based on that, we can now have a really good prediction of DNA flexibility just from the sequence alone. So to summarize the first part, I have shown you some of our data supporting asymmetric nucleosome unraveling and evidence that DNA flexibility governs nucleosome stability under tension. And we are also beginning to map DNA bendability on the genome scale. So Paul, I could actually may take some questions because this is a natural break point for me.
Okay, TJ, there's probably, if you wanted to preserve 10 minutes at the end of questions, we'd have another 12 minutes, but we can take a question now if anybody has one. I don't see anything in the chat yet.
Okay, so let me continue.
Sure.
The second part is now using the DNA in a manner that nature probably never intended, and trying to use DNA to measure mechanical responses of single cells at the single molecule level. So cells have to make important decisions all the time. Cellular decision making is not about, in this case, choosing AT&T or Verizon or T-Mobile. It actually about making decisions based on signals coming from outside the cell. You can have chemical signals or electrical signals. But in the last 20 plus years, we've been learning that mechanical signals are also very important for important decision making by the cells. So mechanobiology at the single molecule level. So we have stem cells and cancer cells that can change behavior based on the mechanical environment. And we also know that they must be sensing the environment through proteins called membrane receptors that are sitting on the cell membrane that are in direct contact with the environment. The question this becomes then, how does a cell collect single molecule mechanical cues and make a decision? So we, in our body, many of the cells that we have are spherical in shape, in isolation. But once they land on some kind of surface or interface, they often spread out. And let me show you a movie here. So cell is landing on a surface. It spreads out just like fried egg, but the cell is very happy and surviving and growing. But if the And this actually is survival in many cell types requires a very stiff substrate underneath, and the cell is able to sense the stiffness through a membrane protein called integrin, shown here. This is a cartoon version of the integrin protein. And through this integrin protein, cells will capture the extracellular molecule and a ligand, and then pull on it and measure the mechanical response. And if the surface is stiff enough, then it'll spread and adhere very nicely, as shown here. If not, if there's a mechanosensing process actually broken, then here's an example of what could happen. So here we have a mutation in the cell that disrupts the mechanosensing. Then you can see that cells initially adhere and spread, but eventually give up and then shrivel and would die. So on the left, that's a happy cell, and on the right, that's a dead cell. The same cell type, except that it doesn't have the correct mechanosensing system. So the question that we can begin to address is when a single integrin molecule senses the mechanical signal, how much force through a single integrin molecule needs to be applied to activate the cell adhesion response by the cell. And if you know the force, then how many integrins actually need to experience that force to start that response. So we developed a method that we call tension gauge tether, or TGT. So ideally, we like to measure the force directly using a calibrated scale, but that was too difficult. So we came up with a different idea of relative force measurement. So imagine having two ropes that can break at 10 Newton versus 15 Newton. And if the weight breaks one tether but not the other, then you can do a bit of a calculation to conclude that the force here must be between the two values, two tension tolerance values. So we are using the same concept. Here we have a cell, the cell membrane, sitting on a surface, and that is the pink one. Sorry, orange one is the membrane protein that is mechanosensitive. And then we have the ligand for that receptor that is tethered to the surface through some tethers. And then the tether is actually a weak, low tension tolerance tether. And so if the cell needs to experience a certain force through the mechanosensitive receptor to activate a certain response, but if that force is larger than the tension tolerance of the tether, then the tether will break, the receptor will not experience that force, and then you will not get a cellular response. But if the tether is strong, high tension tolerance, then the tether will endure, and then receptor will experience high force and then activate the cellular response. So in this case, cell adhesion to the surface. We can actually use the DNA to make these tethers. And here we have a 20 base pair long DNA. And we have a ligand on the top strand, and biotin is a small molecule that binds to is binding partner protein. So you can tether that side to a passivated surface. So when the cell pulls on the ligand through the receptor, then the force will apply. We apply to the DNA tether through that ligand in the unzipping direction. And we know from single molecule measurements that you need about 10 to 15 piconewtons of force to rupture the DNA. So that's a low tension tolerance DNA tether. But if you move the biotin here from the one end to the other end, then when cell pulls on the ligand, then the force on the DNA is applied in the shear configuration, and that requires much higher forces, close to 60 piconewton of force to rupture the DNA. So that's our high tension tolerance tether. You can actually move the position of the biotin to intermediate positions to have tethers that break at intermediate forces. So that is the concept. Then we can have an array of our tethers or TGTs on a passivated cover slip and culture cells in a petri dish. So you load cell solutions and culture them for grow the cells for about half an hour. And then we can gently wash unbound cells and then take photos using a phase contrast microscope, a cheap microscope, and score cell adhesion. And I call this our PN paper because it's almost as easy to use as a pH paper to measure pH, but here instead we are measuring piconewtons of force. So these are 9 different tension tolerance tethers. That's the size of the spot that we put on a cover slip. So with increasing tension tolerance values. And then this is an image of cell adhesion. And as you go to higher tension tolerance value, you can now see a lot of cells remaining bound adhered to the surface. If you go above the tension tolerance of around 40 piconewton, you can quantify this. Below that threshold, very little cell counts per unit area, but above that you see a large increase in cell count, showing that we have a nice threshold about 40 piconewtons to activate the cell adhesion response. So we propose that based on this data and also other kinds of measurements performed in many different cell types, and that there is a universal quantum of force about 40 piconewton to activate the cell adhesion response. So that is the first answer to the first question. What is the force needed to activate that response through a single integrin molecule? So what's going on? So if you have this weak tether, and if you put a fluorescent molecule green dye to the top strand, and then apply force through the tether, then we find that cells do not adhere. You can wash them away. But on the cell area, you see dark patches of fluorescence because DNA tether breaks and through many attempts to rupture the DNA. But if you have strong tethers, cells adhere, and you see a rupture pattern that looks like a ring on the cell periphery, and that's because much later in the cell adhesion process, cells apply very strong forces to rupture the cell periphery. So we then decided to mimic the likely heterogeneous mechanical environment of the cells by mixing the weak and strong tethers in the same experiment. So so these are the 2 different tethers, one above and one below the tension, this threshold of 40 piconewton tension tolerance. And we can use different color dyes to see which is rupturing based on fluorescence imaging. So cell would see an underlying surface with a a mixture of the 2 types of ligands, strong and weak ligand tethers. So if you have a high concentration of the tethers, both weak and strong, as expected, you see a nice adhesion because strong tethers themselves can support a cell adhesion. And we do see this rupture of the tethers in the cell periphery on both channels. And as you expect, if you go to much just the weak tethers only, then cells do not adhere, and we see these ruptures that are more uniform. But interestingly, in the weak tethers, we don't see tether rupturing uniformly. You only see the edge ruptures. So it's as if the cell sees that there are strong tethers, it does not actually apply strong forces to the weak tethers anymore to rupture them in the central area of the cell. So strong tethers alter cells' behavior. The next question was, how many strong tethers actually do you need to alter the cell's behavior. And for that, we kept diluting the strong tethers while keeping the weak tethers in high concentration. And even at 1 million-fold dilution, we could still see cell adhesion. Only when we went to another factor of 10 lower in concentration, we stopped seeing cell adhesion. We can actually do some single molecule calibration and count strong tethers at that transition point, and show that you only need about 1 or 2 molecules of strong tethers per cell needed to activate a cell adhesion, assuming you have a lot of weak tethers. So in a published paper in the Physical Review X a couple of years ago, we proposed ultra sensitivity. We call this Princess and the Pea experiment. So the question is, how many these integrated molecules will need to experience strong tugging to activate a strong adhesion response, and basically you just need 1 or 2 to achieve that. So that's so cell is really sensitive, can actually respond to like small pea buried underneath many layers of blanket here. And we're now using this method to look at some human disease relevant samples. For example, the lung airway muscles that are relevant to asthma, and how the cells can respond to histamine as an allergen, and we can use both fluorescence imaging and force imaging to measure differences between the asthma patient-derived cells and normal cells, for example. So let me stop here. To summarize that I have told you about 2 stories, how we can use single molecule methods to study DNA physics, and how it might exchange biological function of the DNA in the cell. And then second is to use the DNA in an unnatural way to study single molecule mechanical responses from cells. And the work is done by a lot of talented students and postdocs in the lab. I think Reza and Tui were the main people, most recently with Akash. And then Zhufeng Wang was the postdoc who invented the TGT technology on the faculty in Iowa State University. And Myung-hyun is a current postdoc working on the disease-related projects using the technology. Yeah, thank you.
Great, thank you very much, Taekjip, for a wonderful Bell lecture. And I will now turn, in terms of the chat, to open it up for questions. Bianca Granado had a question at the end of your previous section, and I promised her I would call on her to ask it since we have moved on. So Bianca, can you, if you feel comfortable, can you unmute and ask a question?
So I was wondering if you've worked with supercoil DNA and how that would change the different force measures.
Yeah, so that's a really good question. So we haven't published anything yet, so it's still an ongoing project. We can actually make a relatively small DNA circle, but use an enzyme to induce DNA supercoiling, and then perform not mechanical measurements, but mainly fluorescence measurements of various proteins that can sense distortions on the DNA, such as the DNA mismatch. And I think the most recent data that I saw last week, we saw on a DNA repair protein recognizing a base pair mismatch on the DNA. Actually, in a negative supercoiled DNA, you see much better recognition of that mismatch. So I think that is that. So how twists on the DNA can change DNA structure and cause deformations that can be sensed by other machineries is a very interesting question, and now it's clearly within the realm of investigation using our technologies.
Thank you. So if I look back, Michaela, if you feel comfortable unmuting.
Sure. I was just wondering, when you were doing the nucleosome experiments, it's a linear force and the DNA is double wrapped around the nucleosome. So after the first half loop on either side, how did you account for the physical obstacle that was the nucleosome? If I understood it correctly, either the nucleosome would need to rotate so it could continue unwinding.
Yeah. So actually, yeah, that's right. So if you imagine having something wrapped around this cup, and then if you unwrap it, then it'll actually like turn like this, right? So to explain the flexibility dependent unwrapping, actually we did a very simple simulation and changing the interaction energy between the two halves of the nucleosome. But to actually simulate the process, we actually had to simulate rotation of the entire structure after unwrapping a certain number of base pairs. So that's a really good point. Yeah.
Thank you.
Thanks again. And there was a question earlier that I scrolled by, but Kaodu asked me to ask this. It says, thanks for the talk. I would imagine that in each piconewton paper experiment, one needs to use a highly homogeneous DNA sample as the tethers. For example, DNAs have exactly the same number. The question, for example, DNAs have the exact same number of nucleotides as the question. Even that, how would the different conformations of DNA tethers on the PEG surface add noise to the experiments?
Yeah, that's a really good point. So DNA is highly pure. So we chemically synthesize and then goes through a fairly rigorous purification process to have the exactly same length and same chemical structure. But of course, there are some issues that you always have to worry about. PEG surface is what we use to passivate the glass surface, so that our membrane receptors on the cell membrane do not stick directly to the surface. They only interact with the ligand tethered to the surface through the DNA tether. But that's not always perfect. So if there are some rough patches on the surface that is sticky, then that can also compromise experiment. So for example, DNA can also melt, right? Only we have 20 base with DNA. Sometimes DNA can break into two component strands and then directly to the surface, forming essentially a unbreakable tether, extremely strong tether that can also cause some issues. The fact that we see this ultrasensitivity, that we only see the response when you have some amount of strong tether, suggests that that is not such an important issue here. Yeah.
Okay, thank you. And David Juncker, can ask at least one of your two questions.
Oh. Hi. I muted myself. Thanks. Can you see me okay? Well, I think that was a wonderful talk. Well, if I get to ask one, maybe I'll ask the second one about catch bonds, which strengthen upon tension. I don't think that's something that is the case in your DNA. So could you comment maybe on how that might affect the results you're seeing with these edge effects, or whether you expect that's also something, I mean, that's something that happens, or maybe that catch bonds could address, and whether that's something that could also potentially be built into your sensors?
Yes, so DNA does not form a catch bond as far as I know, but integrin protein actually forms a catch bond with the ligand. And in fact, there is a paper by a lab in Georgia Tech showing that, but also there's additional data that looks very interesting, that if the integrin protein requires certain force to get activated, right? But once you apply force to activate this, then even after the force is gone, it stays active. So it seems to remember its past experience of high force. And that actually may be part of the reason why we see this ultra sensitivity in the Princess and P experiment. And but yes, another related question from you is that, can you actually make DNA structures that are smarter, right? They can respond and change their responses according to the external environment. So that something that we are also looking into. Yeah.
Okay. Thanks.
And David, I've been told that we can probably go a little bit longer in terms of questions, so I'll circle back to you for your second question, okay? So Paul Francois has a question.
Yeah, hello, can you hear me? Okay.
Yes.
Thanks a lot for this great talk. Yeah, I had a question about the P and the Princess experiment. So I'm interested in immunology, and immunology, what you have is that you also have molecular sensitivity to very strong ligands. And this comes at the price of essentially antagonistic reactions. So because you don't want weak ligands to conspire to basically somehow be considered a bit like a strong ligand. And so I'm wondering if you had some similar effect for adhesion, like is it a purely additive effect, or could you also have antagonistic reactions? So for instance, middle strength tethers will kind of prevent adhesion in some way, which I think is related to the spatial aspect. I think in B cells, some effects like this have been observed. So that's why I'm wondering a little bit.
Yeah, so that's a really great question. For some immune cells, I think there's good data to support the idea that immune cells may respond to even a single ligand, and so we actually try to directly count how many strong tethers are under each cell. So I think I have one slide here, so we can actually see. In this image, under each cell outlined by the yellow, and you can see strong tethers, and then you can see, okay, 1, 2, 3, 4. I think I have, yeah. So then we can actually plot the histogram of how many strong tethers we actually see under each cell, and instead of having like exponential distribution, we see a nice peak at 2. So it's not a princess and the P, it's princess and 2 P's. And so the way I think about this is that you need to have, if you want to actually pitch a tent, and then so that you can walk freely under the tent, then it's going to have two tent poles. Then you can have a local nucleation that can propagate along a line to achieve this. But
very interesting. Thank you.
And TJ, I'm going to take the liberty of following up on that. In terms of the strong tethers, have you looked at anything in terms of the kinetics of the downstream after that, in terms of recruitment and possible communication between these different centers?
Yes,
recruitment of other adhesion molecules and things.
Yes, so now we are doing like 3-color experiments. So we are actually using very good antibodies against the integrins, both regular state and the activated state. So we can use one color to do TGT rupture, and then we can use a second color to image integrin activation, and third color to image recruitment of additional integrins to the same site. So we can see what happens a little downstream of integrin activation signaling.
Very cool. Very cool. Great. And David, I promised I'd circle back. I think that's, you get to ask the last question.
Okay, well, thank you. That's quite a responsibility here. So, we actually in our lab we have also worked a bit on patterning, nano patterning, down to maybe 200 nanometer in size of these spots. Where there was been quite of work in the past of like trying to see the minimal size of these patterns, but based on your work, it seems like just 2 molecules are enough. So it's quite remarkable. And yeah, is there an interest or is there a benefit in maybe patterning some of these nano pattern DNA patterns?
So I think we need maybe you need 2 strong molecules, but we need thousands of weak tethers. So we cannot just have 2 strong tethers. We need to have lots of weak tethers. Otherwise, cells cannot adhere. Right? Just two attachment point is not enough to have a nice adhesion. So I think patterning is still very useful because you can pattern the arrangement of the majority of the tethers. So I think in the past, we also tried to do some patterning experiment in collaboration with a lab in France, but we haven't quite finished that. So if you like to try something together, we can chat.
Well, happy to talk. Yeah. So and maybe one is the strength of the strong tether. Did you also play with that? If you make it a little bit weaker, will 2 still be enough, or then you need many poles?
So that's another really strange thing that I didn't tell you about. That part we haven't published yet because it's still hard to really believe. So if you have 2 weak tethers, but both under the 40 piconewton threshold, it still works. So let's say 12 piconewton and 23 piconewton mixture, that still works. I think what's going on is that when the weak tether breaks, and then there's a stronger tether that's remaining nearby, then the weak tether can be formed because you are still pinching down like a membrane there, and therefore the broken tether can be formed very soon. So actually, making the tethers actually effectively stronger. So that is our thinking.
Well, thanks so much.
So cell is doing the differential force measurement, not the absolute force measurement in that sense.
Well, fantastic. I think given a lot of things to think about and very exciting work and very exciting what's coming, I'd like to ask everyone to join me in thanking Professor Ha for giving us such a wonderful Bell lecture this year. And glad you could present it remotely, even though we couldn't host you directly, but I look forward to when we do get to host you back in Montreal. Thanking you again, TJ, and wishing you and your family all the best in terms of health and stuff. Thank you so much.
Thank you. Thank you so much.